Hi, welcome to another episode of Moment of Science. I'm Cameron Sahami,
and I'm Kachunyu.
And today we have a very special guest with us, Dr. Michael Grant from EPOB Biology at the University of Colorado. And Dr. Grant is going to talk to us about evolution. Could you introduce Dr. Grant?
Yes, Dr. Grant got his PhD from Duke University in botany back in 1974, and he's been affiliated with the University of Colorado ever since. And his research interests are in evolutionary theory, computer simulations of evolution, as well as forestry genetics. And he's gotten many teaching awards in the past, and last year he was the recipient of the Hazel Barnes Award for Teaching and Excellence from CU. And this year, I think he was nominated for U.S. Professor of the Year. So welcome, Dr. Grant.
Thank you. Glad to be here.
And I think evolution is quite topical now, especially in Colorado, since just last year there was a big hubbub in Jefferson County where a high school student, Danny Phillips, I think was his name, who basically caused a big deal with the school board, and he was quite offended by the concept of evolution being mentioned in a video that the class showed. And I think there are lots of misconceptions about evolution in the public, and I think you're going to hope to clear up some of these today.
Well, I can talk about some of the things I hope that will be of interest. Mm-hmm. One of the misconceptions that I think is most important is that when we talk about evolution, we want to talk about science. Mm-hmm. And when we talk about science, we have certain rules and patterns that we have to adhere to in science, and that is distinct from religious studies. And our view is that we should do science in the evolution class, and we should do religion in the religious classes, and keep the two distinct.
Mm-hmm. Mm-hmm. Well, let's go ahead and get started with the slide presentation. I think we've talked to you a little bit about this before, and I think these slides will be quite elucidating.
Okay.
So if we could have the lights off in the studio, please, we can go ahead and get started.
Get the lights?
Lights?
Well, it would seem appropriate to start a little talk on evolution by mentioning Charles Darwin. Obviously, he is the individual who did the most to generate the ideas of evolution. We have extended those ideas considerably beyond his time, and we know a fair amount more about it than he did. And so it's not a matter of being static, as most sciences are. It progresses pretty regularly. But his book, The Origin of Species, is considered to be one of the three most influential books in all of mankind that has been produced. And in particular, the idea that he has, this idea of all living organisms being related, is a very different way of looking at the world than had been up until his time. As he said, there is a rather grandeur to this view of life, and it's one that I subscribe to. Next slide, please. We talk about the Darwinian theory, and oftentimes we'll hear things that say it's just a theory, meaning something that it's a guess or no more than an idea. And I would argue that the Darwinian theory, the theory of gravitation, the theory of electromagnetic fields, the Copernican theory, all fall in the same category. The theory is basically what most educated people believe to be true and correct, and backed by a large body of supportive evidence. That's what we have to do to reach the status of theory, and the Darwinian theory certainly reaches that status.
Yes, so the scientific definition of theory is very different from a colloquial definition of theory.
Absolutely. But colloquially we'll say theory meaning I just have an idea or a guess. And we work from a guess, an idea, to a working hypothesis, to a hypothesis, and it really takes an awful lot of supporting information before something really qualifies as theory. Next slide. I wanted to talk a little bit about the way in which we understand the evolutionary process, and can we see it in ordinary terms, and can we see it in real terms? And the answer is yes, we certainly can, and one of those places are in what I call natural experiments, experiments that have occurred in nature, and in particular we can demonstrate and recognize the evolution of chemical resistance. Insecticides, herbicides, and antibiotics are some of those that are most important. Next slide, please. This slide, I think, is really quite illustrative of the general notion of natural experiments. On this axis, we have a measure of the resistance of houseflies to a chemical that didn't exist earlier than the 1940s, and that's DDT. DDT then became widespread in the environment, became a part of the environment, and you can see back in the early period, the resistance scale was, there were very, almost none, but a few flies that were resistant to DDT. And as it became a more ubiquitous part of the environment, we have an evolutionary change, a trajectory, such that the resistance to DDT increased very dramatically over a very short time span. We're talking about a half a dozen years here. We're not talking about 100 million years. And this is an evolutionary response. It is a genetic response. And so we can see evolution in action and evolution in operation in this case.
So what's happening is that the flies that were resistant to DDT live on to create other generations of fruit flies.
What happens is that the flies that are resistant reproduce more successfully than the flies that are less resistant. It's not an either or, it's a continuum. But the more resistant the flies, the more they reproduce successfully, and those genes for resistance then are passed on in greater frequency to the next population. So reproductive success is the key in an evolutionary context. Next slide, please. This is a very similar kind of experiment, except this is with mosquitoes, and this shows on this particular axis, this is the percentage mortality. So at the start of the experiment, the mortality rate in the presence of this amount of DDT was nearly 100%, about 99, 98%, so only about 1 out of every 100 mosquitoes survived. Left the DDT in the environment, and after about 10 months, then a little over half or almost half were surviving, and after only a year and a half, then 20% mortality is where we went. Very rapid evolutionary change. Again, this was in response to DDT, a natural experiment. It wasn't contrived, we just had DDT in the environment, and this was the response. Next slide, please.
So that's sort of evolution in action, basically, over the course of a year.
About a year and a half in this particular case. Now, those are natural experiments. This is a contrived, artificial experiment. We can use the principles of Darwinian evolution to operate in the laboratory. This is percentage of oil content in corn seeds, and these are selected lines that started at about 5% of oil, and this is a down-selected line, so individual corn plants that had the lowest amount of oil were bred together and then harvested and done again and again over about 80 generations here. This was those that produced the highest oil content, and you can see that over about a period of 80 generations, the oil content was increased from about 5 to 19 units, almost a 400% increase, using the idea of Darwinian selection, except in this case, instead of nature making the choices, humans made the choices. Artificial selection. Next slide, please. Okay, now one of the things about evolution, the idea is that all living things are related. And for all living things to be related, we have to connect them in some sort of way. And one of the ways that we can think about connections as humans is a genealogy. And so let's start that as a concept. And next slide, please. That we can think about brothers and sisters and moms and dads being related, and we're talking about genetic relatedness, and the details are not really important to us here. But these form a group of highly related individuals. Then we have an extended family, nieces and nephews, aunts and uncles. They are also genetically related, but less so than the brothers and sisters. And then we can extend out to great-grandparents and first cousins, and we can make nested boxes larger and larger. These all show a relatedness, a connectedness to each other in a hierarchical, inside to outside fashion. Next slide, please. The same idea goes to phylogeny. Phylogeny is the term we use to describe evolutionary relatedness, but it's the same as a genealogy, except the scale is a bit different. May I have the next slide, please? If we have a series of critters, and I just took a whole series of very different kinds of critters, whether we're talking about a moth, a human, a carp, these are a set of organisms that we could ask, what are their relatednesses? How do they fit together? Which one is more like which? Next slide, please. And most people would argue that this is a fairly reasonable connection, that the donkey is more like the horse than either of them is like a chicken or a turkey. but a chicken and a turkey are more like each other than either is to a carp or a tuna. And so now we have the next level. We moved out to the first level of connectedness. Next slide, please. And now we can put things together in slightly larger categories as the extended family gets larger and larger. Next slide, please. And then we can begin to group these things in the boxes just as if we do in genealogy. So these are most closely related. Then in the next larger box, they form another group. The next larger box is a more inclusive group, and so on and so on and so on. So we have a connection between, say, the human, the chimp, and the rhesus monkey, all the way down to the euglena, a single-celled organism that most of us don't think of as cousins very much. This is the idea of phylogeny, trying to determine the connectedness. Next slide, please. And this is what a more formal phylogeny looks like. It's a little bit more intimidating. And let me take just a little bit of time to show you what we've got on this axis. This is a phylogeny, a relatedness diagram that shows how different organisms are related to each other. And on this axis is estimates of how long ago the group diverged. And that's based on fossil records and other kinds of evidence. This is mostly molecular data, which looks at the degree of similarity in molecular constituents amongst different kinds of organisms. And if I might point out 2 or 3 little things, there's a little group of organisms right over here. See, they're connected at that particular point, which means that that's a box. We put a box around that, and in that box we have the pig, the sheep, the cow, the horse, and the donkey. That forms a box of that particular size. Now there's a smaller box right here that's the horse and the donkey, as we started out with. So we go from the horse and donkey to the smallest box up to the next largest box. And then we can move back up further and further as we go out and extend the family to larger and larger groups of organisms. There's a nice little box that you can see right over here, a little collection right in here. And those are a series of plants, and those are different relatednesses amongst the plants. But notice that the plants, if we go back far enough, are also connected over here to the pig and the sheep and the donkey.
And this sort of hierarchy here, the connectedness of this sort of superfamily tree, is borne out in fossils?
Well, certainly there are many consistent relationships between these and the fossil records, particularly if you go to the higher levels and look at such things as the origin of the mammals, the origin of the birds, the origin of the flowering plants. If you try to look at individual species, it's pretty hard in the fossil record, especially for a lot of these which don't fossilize really very well. But there is a consistency between the two. There's not perfect agreement in all cases, but there is a high level of consistency between them. The fossil record is one of the pieces of evidence that we use to give those dates. The other is the degree of molecular divergence. So the further apart the molecules are, the more likely the common ancestor was further back in time. And so here's our euglena, way over here, about as different an organism as we can think. And in order to connect the euglena, we really have to go back as far as this particular phylogeny can take us in order to connect the euglena with the other kinds of organisms.
So that was at least a billion years ago.
Yes. These are millions of years ago, so that's 1,000 million years ago. If you're British, that's the way you'd say it. Okay, now we can do these phylogenies with many different kinds of data. We can do it with morphological data, behavioral data, distributional data, and all kinds of molecular data, nucleic acid, DNA data, RNA data, proteins. And the more of these lines of evidence that we put together, and the more times they're consistent, the more confidence we have that these relationships indeed are correct. And the more we do this, the better the patterns seem to fall out. Next slide, please. Now, one of the things that often comes up in discussion of evolution is the ideas of intermediates. And I put this slide in because it's a pretty clear indication of what someone's notion of an intermediate might be. We've got a lizard head, human hands, we've got a dorsal fin, tail. One of the things that comes up is people say, well, we've never seen one of these. If man evolved from fishes, how come we don't see this? Well, what we conceive of as intermediates, we as humans and can imagine, may have very little to do with the actual evolutionary intermediate path that evolution followed. It made its own choices and its own pathways, and they may not follow, and in fact generally do not follow, what we can think of as what the intermediates ought to be. Next slide, please. This is a much more realistic notion of what an intermediate is. This is a walking fish, and it has these fin rays here are much thickened, and it can actually leave the water and trek out across the mud flats for a distance, looking to expand its range and to colonize new habitats. And so this is the kind of intermediate that is much more defensible and understandable evolutionarily from a exclusively aquatic to a partially terrestrial to aquatic kind of organism. So that's much more what an intermediate might look like. Next slide, please. The idea of intermediates is really quite important, and I wanted to take a few minutes to talk about one of the most common things. Is that the idea is that we have, for example, the mammalian eye takes all the pieces to work. That if you poke out one part of the eye, the rest of it doesn't work. You need the whole gizmo in order for it to work. So how could a partial eye evolve? How could getting half an eye be adaptively significant or evolutionarily advantaged?
So how could a simple mutation ever give you this very complex organism that you need as a functional eye?
That's right, and that's another way of saying the same thing, that if you start from no eye and then you jump all the way to a complex eye, how could that happen by macromutation? Or if it didn't jump that big, if it only jumped halfway, that wouldn't do it any good.
Yeah.
So let's look at that a little bit. Next slide, please.
Interesting.
Okay. I have a series of, quote, eyes that are existent now in mollusks. I use mollusks as an example because they give us the full range. So each one of these examples I'm going to talk about exists. We don't have to think of it. We don't have to imagine it. We don't have to look in the fossil record. We can look at living organisms.
Wow.
There are mollusks that have a thin layer of epithelial cells. Those are surface cells, some of which form pigmentation and have a few nerve fibers. These pigmented cells can respond to light and dark. And that's all. They don't form an image. They don't form any other visual cue, just light and dark. Light and dark can be a matter of survival to mollusks, and is a matter of whether one is protected underneath a rock or one out in the open on a sandy bottom or something of that sort. So that's much less than half an eye. That's much more like 1/100 of an eye. But it is functional. It matters. It's a light sensor.
And it gives it reproductive advantage.
And it makes a difference because it is a information gathering system for the environment, and that information then can translate into reproductive success. There are another set of mollusks that have these pigmented cells formed in an invagination, in an indentation. And that indentation increases the surface area. And by increasing the surface area, it increases the sensitivity. So shades of light can now be taken into account more effectively. Another mollusk has this indentation enlarged. It's larger, but this is still water-filled cavity. Most of these are ocean-dwelling organisms. Still has the epithelium, but the optic nerve is now developed really quite extensively. This is the pigmented layer through here. And so this is a substantial advance over this in visual acuity. Then we can go to the next level, which has the similar kind of shape, the optic nerve, but here this little opening has closed, and the cellular fluid here is produced by the organism, and that cellular fluid can be produced optically in such a way to make it more efficient. Then we have the next stage where we have the fluid inside. We can form a lens. Then they form a cornea, and then in the most complex eye, we have all of the pieces that we would think of in a vertebrate eye, with the lens, the iris, the cornea, and the retina. So you can see just in those 6 categories how one goes from much less than half an eye to a full eye in moderate steps that are evolutionarily reasonable and plausible and easy to understand. Next slide. One of the keys to reproductive success that I wanted to talk about, one of the keys to evolutionary biology is reproductive success, and I want to give a couple of little examples that I think are fairly interesting. This is a scorpion fly, and scorpion flies, the males their reproductive success is determined by the females under female choice about how good their food supply is that they offer the females. This has the anthropomorphic name of nuptial offering. The males offer this to the females, and the females decide on which males they'll mate with based on the weight and therefore the quality and the nutrient in that particular offering.
Whoa.
Well, one of the things that has happened here is that there's a group of male scorpion flies that have developed the behavior of taking, once they capture their prey item, instead of taking it directly and offering it to the female, they take it to a local water puddle and soak it in the water puddle so that it increases the weight, and then when the female assesses it, it's of higher quality than it was before. And so the males that have this behavioral gene for dipping it seem to have a higher reproductive success.
I hope this doesn't give any of our viewers any ideas.
Okay, next slide, please. Now, there are a number of other cases where you can see this reproductive value illustrated in a very interesting sort of way. And I mentioned the scorpion flies, and let me talk just a minute about the bed bugs. Bed bugs have some really interesting reproductive patterns, one of which is they engage in what's called traumatic insemination. That means that the male bed bug actually injects the sperm through the wall, the cavity wall of the female, and that's why it's called traumatic insemination. There's little fuzzy patches that form on the sides of the bodies of the bed bugs, and one of the interesting things is that in one of the species, the males have developed a patch, and that patch stimulates other males to engage in the traumatic insemination. So a male will actually inseminate another male. Now, evolutionarily speaking, why? What sense does that make? Well, for the male that stimulated the insemination and received it, that male gained some nutritional advantage. That is, resources are very much in scarce supply in nature, and so that gained a nutritional advantage. And the second advantage, it disarms the other male, and there's competition for mates. And so there are two advantages to that, getting one male to deceive another male into inseminating the wrong kind of a bed bug.
Let's hope none of our viewers get any ideas from that.
Well, let me tell you one more thing about the bed bugs, that there is also a trait that in some of the bed bugs, where the male has been inseminated by another male, that the male that actually contributed the sperm, male A contributed the sperm to male B, male A's sperm will actually be motile and move into the reproductive tract of male B. So when male B mates with a female, he will be contributing sperm from male A and not his own. And I think that's about as extreme as one can get for that sort of thing. But the point of all of this, and there are interesting stories about all of these of that general category, is it emphasizes that the success in evolutionary terms is reproductive success. We always hear the phrase survival of the fittest. Darwin never used that phrase. It's differential reproductive success. Of course, survival is part of it. If you don't survive, you don't reproduce. But the converse is that a lot of organisms can survive and still be evolutionarily different. And okay. That's as far as I'd want to go on this right now.
Let's see if we have some calls lined up. If we could go ahead and studio lights lit up. That's really interesting. So the idea here, I can move that chair up a little bit, is that reproductive success is the key.
Yes.
Whatever will give a particular species reproductive advantage, then that is the mechanism by which evolution moves forward.
It's the reproductive advantage to the individual, not the species. It's not the group. So it's every individual is operating to maximize their own individual reproductive success. As a consequence, the species may, and in general, will be advantaged. But the idea that it's the species that's being advantaged is a particular technical argument called group selection. In general, that's thought to be a fairly weak force, that it's the individual that is the key.
Okay, I think we do have a call.
Yeah, if you go ahead and pipe the call in here, please.
Yes, Dr. Grant, this has been fascinating so far.
I can't quite hear that. Could you check the phones and...
Okay, Dr. Grant?
Yes.
Okay, you hear me now?
Yes.
Yeah.
This has been a fascinating show so far. Now, it seems to me, I don't know how long ago this was or where I saw the reference, but it seems like every 12 to 20 million years there's a major evolutionary change or jump through Mother Nature, not otherwise externally caused. And it seemed like we've gone a long, long time without having any such change. Is that true, and is there any thoughts on why?
That is one of the things that people argue quite a bit about. There's the nature of whether there's periodicity to extinctions, And when you have major extinctions, oftentimes, well, generally, you will be followed by major periods of speciation, formation of new species in those particular cases.
Sort of to fill the void?
That's part of the reason that there is space in the ecological sense available, and the competition is reduced, the food supply is increased, and you get increased numbers of species following major extinctions. And a couple of guys, Sepkowski's one, that says those occur maybe every 26 million years, driven by some sort of external force, obviously. And he has some fairly decent evidence for it. The problem, of course, is trying to get periodicity over 26 million years per period takes an awfully long time span. And there's quite a bit of disagreement about whether it's real or apparently real. The predictability about whether we're going to have one next or not is something that I certainly have no way of knowing. I don't know whether we're in for something rather large or not. If, for example, we had something like at the Cretaceous where we had the meteor that struck the Earth, that certainly caused a major resetting of the biota, and certainly would happen again today. And I don't know whether we're expecting that or not. I don't know anything.
Now back to this idea of speciation, I think a lot of people who...
Thank you, caller.
Yeah, thank you, caller. Who don't like the idea of evolution, one of their arguments is that scientists haven't really seen any evidence of speciation going on today. Now, do we have evidence of that?
Okay, I'd give two answers to that. First of all, any look at the fossil record shows we've had millions of species arise. The flowering plants were not around in the Devonian. Neither were the mammals or the birds. And all of those species didn't exist at one point in time, do exist at another point in time. There's not any other conclusion one can arrive at except they have arrived. The other answer I'd give to that is that we can generate artificially in the laboratory new species. Plants are the easiest to do that with. Perhaps one of the most famous examples is primula. Take two species of primula that won't cross with each other. Under artificial lab conditions with the right sort of chemicals, you can get them to cross and double their chromosomes. They produce a new species that has twice as many chromosomes as either the parents. The new species crosses with itself, produces viable progeny, but will not cross with each of the other two. That's the definition of what a new species is. So we can, and there are a number of other examples as well. So we have done it artificially, and it does take place in time spans that we can look at it under artificial conditions.
Great. Well, I think that's going to do it for our time for this particular episode. I want to thank Dr. Grant for being here, and I want to thank the caller for their great call. If anyone is interested in these topics, you can check out our webpage, and I think the address is going to be coming up in just a second. And there will be links to Dr. Grant's page and some various other evolution sources that you could take a look at and pursue this idea a little bit further. June?
Yeah. All right. Well, thank you very much again for appearing on the show, Dr. Grant.
Yeah, thank you.